First of all, I would like to thank the organizers for giving me the opportunity to speak here and present our work that we published last year. And I have to click here, right? So what I want to present to you in the next 15 to 20 minutes is how you can use sequence data alone to predict protein-protein interaction interfaces, the structure of a protein complex, or even predict how several subunits in a large complex interact. That is interesting for a wide variety of fields, not only to understand cell function, but also to apply it to drug development, because basically everything in a cell works through teamwork, interaction of any kind, and protein-protein interactions are important for aspects such as transport across the membrane or signal transduction. And so inhibiting or stabilizing protein interactions can be a very interesting method for whatever basic research, but also, as I said, for drug discovery. So far, most methods rely on experiment. Well, most of the time, when you want to determine something about protein-protein interactions, you have to rely on experimental methods that are often time-consuming, tedious, and low throughput. So the question that we asked was, can we also use the sequence data that's currently coming up at a very high rate across multiple species, and can we use that to infer or make predictions about protein-protein interactions? There actually have been quite a few methods that use available data to make predictions that ranges from machine learning to homology modeling, considering similarities between the phylogenies between the two proteins of interest, but also using covariation. And covariation is basically saying that if you have two proteins that interact, then if there is a mutation in one, does it work? If there's a mutation in one protein, there has to be a compensatory mutation in the other if those two residues are indeed interacting or in the binding interface. And there are a few people who did that already more than 20 years ago, but had issues with availability of sequences and transitive contacts. And then a few years back, another group of people showed that you can actually use a global model instead of just looking at those positions independently to get rid of transitive contacts that you predicted before. So how does it really work? In single proteins, but also complexes, what you can see is that those residues that are close to each other structurally usually also co-evolve. So you can see a trace in the evolutionary record, so you see co-conserved columns, and if you know that those residues are close in the structure and you look for them in the sequence alignment, you indeed see that those co-evolve. If you want to go the other direction, it's much harder. So if you just have the sequence alignment and you don't know what you're really looking for, then inferring the correct context is a bit harder. And that has been shown to be possible, though, a few years back, about 4, by using a global statistical model. So what you can do is actually you have the multiple sequence alignment, all kinds of different organisms, and you look for all these columns, and you look which columns basically co-vary, and then you build a global statistical model. So you consider those two columns, for example, those two green ones, in the background of the full multiple sequence alignment. From that, you get scores for each possible pair of residues. You can sort them by their strength, and if you wanted to draw them on a two-dimensional representation of the structure, you could do that here. So this would be residue 29 and 61. And then if you use those as constraints, for example, for protein folding, then you can reconstruct the structure. You can read that up here. So if you want to do that for protein-protein interactions, that's what we did, then you need to have one multiple sequence alignment and not the two individual ones that you would easily get for your two proteins of interest. So instead of doing what you can do for one protein, we just want to do that for two proteins together. And for that, the first thing is we need to pair up the correct protein sequences that we have for the two interacting proteins. And once we have that alignment, we can do, for our global model, get couplings not only within each of the molecules, but also between, which is illustrated here in this quadrant of the contact map. And with those contacts, you can use that for as constraints in protein-protein docking. There is whole bunch of software available to do that. And once you have the docking, once you have the protein, the monomer protein structures and the constraints, you can reconstruct the full complex. There are two things that you have to consider. One is the alignment concatenation. As I said, that's the most important part, because if you put in wrong pairs when you infer the model, when you create the model and you create it based on wrong concatenated sequences, what you get is a whole lot of noise, because you basically say, well, this paralog, I think this interacts with that paralog. But if it doesn't really, what you get as context will be wrong. There are multiple ways you can do that, and there are, for many proteins, we just don't know how to concatenate them. But in bacteria, for example, it's pretty easy, because you can just say, first of all, we pick the closest homolog, and if we have multiple and we need multiple, we want to include multiple because we know the complex has multiple paralogs in each species. We can also look at the genome architecture and say things that are on neighboring operons, or even on the same operon, are more likely to interact than those that are very distant in the genome. The second thing that you need if you want to do predictions about proteins and actually compare them is that you need a score to know where to stop, to know when those couplings that we predict are actually true positives or when they are actually just noise. So what we developed was a score that basically considers the background distribution of the score coming from the global model that we use. And then also, because the score distribution depends on what we put in, namely the multiple sequence alignment, is also we have to normalize for the size of the sequence alignment. That means if you have many sequences or a very, very large complex, then the score distribution is still centered around the zero mean, but the absolute values can be very different. We did that, so we developed the score, and then for a bunch of like 20 complexes as a set, evaluation set or training set to some extent, we had a look at what score threshold we would get a sufficiently high precision, so we could use the score and say everything above that we trust as true interacting couplings between two proteins. And looking at a bunch of proteins, we got a threshold of 0.8 that we used for our method. We then evaluated that based on a large data set that we got from the literature with about 3.5 protein-protein interactions in E. coli. Because we do the concatenation based on genome distance, we filtered that data set for only those complexes that are actually indeed on the same or neighboring operons. We lost most of those complexes in the data set due to that. We then created the monomer alignments, concatenated those as I have shown you, and after creating the alignments, we built the model and calculated those coupled residues within and between the proteins. In the data set of those complexes that were still left, we have those that have a solved 3D structure in PDB, and we used that as evaluation set. And then we had those that didn't have a predicted 3D structure, and for those we just predicted the couplings as a prediction of the binding interface. For 15 of the complexes that had a known 3D structure, we also performed protein-protein docking using HEDOC. So we tried to actually reconstitute the extra protein complex. So what are the results? In total, we got about 2/3 of all the predicted pairs that had a score, a normalized score above our magical threshold of 0.8 to be correct. Correct means here that we defined everything that is closer than 8 angstrom in the protein complex as a contacting residue pair. We also were able to reconstitute the binding pose, the correct binding pose, with an interface RMSD of less than 4 angstrom for the 15 protein complexes that we docked. The whole method actually works in quite a fast manner, so predicting and building the model, predicting the coupled residue takes a couple of hours for a medium-sized complex, which means like 500 residues. And if you want to create the models, because you have the constraints coming from the coupled residues, you can actually get along with like creating about 100 to 200 models to get the correct pose. So as I said, for 15 of the complexes, we performed docking. And this is one of the examples. It's basically the example belonging to this contact map here. So let me just walk you through this first. What you can see here is is an exemplary contact map. A contact map is a 2D representation of a 3D complex. So the rows and the columns are the residues, and you see a gray dot everywhere where there was a residue pair that was closer than 8 angstrom in the complex structure, the soft experimental structure. And the colorful dots are the residue pairs that we predicted with a score above 0.8. What you can see here already is that there are a lot of green dots on top of the gray dots. So basically the couplings that we get from our model coincide with a docking with the residue pairs that are actually close on the interface between methionine transporter subunit I and N. So when we use those green constraints here, so if we use those residue pairs as constraints for docking, we get a model, which is here shown in orange and purple. And if you overlay that again with a soft structure that we used to make the gray dots on the slide before, you see that they coincide very well. And if you want to quantify that, you get an interface RMSD, so that's just an RMSD over the residues that are at the actual known protein interaction interface of 1.2 angstrom. But even if you create a full C alpha RMSD for the complex, in total you get a very low RMSD. You can also see here where we compared in for docking, we didn't just dock with our constraints, we had a negative control in which we didn't put in any constraints. And that would be what you can see here in gray. So we had just used the Haddock toolbox without any constraints apart from center of mass. So we allowed the complex the subunits of the complex to touch, but we didn't put in any constraints about where the interface lies. And the blue dots here are the ones where we put in the predicted interface residues as restraints. And what you can see here is all of the 100 models that we created with the EV couplings as a restraint are very, very similar to the experimentally solved structure, whereas none of the ones that were created just by center of mass. Given that this worked quite well for all of our complexes in our evaluation set, we also asked the question whether you could use the score to predict which subunits in a larger complex interact with each other and which ones are just involved in the complex but not physically touching. And for that, we used ATP synthase as an example. Here you can see an example or illustration of what people think ATP synthase looks like as a full complex. And if you take all the subunits apart, so alpha, beta, epsilon, gamma, the C ring, A and B, delta, you can make a triangular matrix of all the possible pairs of subunits. So what you can see in the upper part of these triangles, in the left triangle basically on this part here, is the knowledge of which subunits interact from literature. So we went through the literature and looked, what do people know already, which subunits do interact physically and which ones don't. The triangles on the bottom here are the interactions we predicted based on the EV complex score. So everything that is blue in the top part is known to interact from literature. Everything that's blue in the bottom part is predicted to interact based on having at least one residue pair coming from the global model with a score of above 0.8. What you can see is there are a few pairs where either the literature is not conclusive, so they're contradicting information, or our score was just below the threshold, so we don't really know if this is just a question of calibration of the score or if it's false positive prediction or false negative prediction. In total, we got 85% of the possible subunits correct, so you can conclude that you can indeed predict the truly interacting subunits in a larger protein complex based on this evolutionary coupling data, which just comes from sequence information. So you only need sequence information and you can predict which subunits in a larger complex truly interact. As I said, we also did a few predictions for all those complexes that were in our initial data set that don't have a solved structure so far. And one of these complexes, the toxin-antitoxin complex DIN-J-F-Q, was solved experimentally and published while our paper was in review, so we could actually evaluate that prediction on the fly. And what you can see here is that 95% of those couplings on the interface that we predict are actually true positive coupling. So they do represent the interface. Same applies to the couplings within the two monomers. You can try that method yourself if you want. You can just go to evcomplex.org. You can actually also try the protein monomer folding prediction there. So you can go the full way of predicting monomer structures from sequence data, and then also predicting complex interactions from sequence data. And then if you put that together, you can get the full protein complex from sequence data alone. So in total, I hope I just showed you that you can use, as I said, sequence data, the evolutionary record of two proteins together to make predictions about where the interface lies on these two structures and if those two proteins indeed interact. There is currently still a limit of sequence data that is available, or sufficiently diverse sequence data for a whole bunch of complexes, but for most prokaryotic complexes that we looked at, there was indeed enough sequence data available. You can also extend that method if you know how to do the pairing up of the sequence pairs, the two protein subunits basically, or the two proteins in the complex. You can actually extend that to, for example, host pathogen interactions or protein RNA interactions if you wanted to. And last, I just want to give a big shout out to my co-authors that are listed here. All of that did a tremendous amount of work, and I would thank you for your attention. And if you have any questions, I'm happy to take them.
Yes.
So my question is, in protein-protein interactions, sometimes allosteric interactions are very important, and some residue could co-evolve that actually are not in direct contact with each other. So how do you filter out this kind of signal, if you do it?
Good question. So at the moment, there are not really many ways of filtering those out if we don't have any structural information, just because they all appear similarly co-evolved in the complex. What we do look for, if, for example, we have the subunit structures individually, and we see that those two complexes, the couplings between those two proteins fall onto really two very different binding interfaces. So if you look at them and they're really on the opposite sides of the protein, that's a strong sign for them being allosteric or like alternative conformations. But so far we don't have an automated way of disentangling those.
Yeah, thank you for very nice talk. In the end, you said you could use it for host pathogen protein-protein interactions, and I'm just thinking intuitively that wouldn't work because the host would maybe try to evolve away from the pathogen. So I'm wondering, have you tried that already, and do you have any proof that that would work?
No, so that's basically one of the ideas we're trying to pursue in the future. And there are multiple problems that actually come up with that. And one is, as you said, the host tries to evolve away, but then actually the pathogen tries to evolve after that, right? So you would have the coevolution. But then the question is, how do you really pair those partners up? Because usually you don't have one pathogen that only affects one host organism. You usually have some that are across species. So, and that's basically the main drawback at the moment. So if you find out how to pair those up, you can, in theory, use the method, but that's the harder part.
Thank you for your presentation and also for your continuous work on evolutionary couplings approach. I was wondering, as you mentioned, the number of sequences appears to be the major constraint currently. Do you do any work in order to improve the method so that we would need less sequences? Because I imagine that, as you mentioned too, for eukaryotic complexes, you probably cannot collect enough sequences because the proteins are long and because the organisms are just not diverse enough. So do you do any work in that direction?
We are to some extent, but with the global model we are currently using, there's just no way of reducing the number of sequences that we need to make statistically significant predictions, right? So in that direction, it's really hard unless you change the method drastically. But as far as I know, there are approaches where people try to methodically mutate sequences and still see if those interact and use those then as an extension to the alignment. But I'm not involved in that personally.
So obviously the sequence alignment is going to be very important here. What is a good method that you've been using?
So we're using Jackhammer, and we are also sometimes using HHBlitz, so we're happy with both of them. The problem with HHBlitz was that the database hasn't been updated in a few years. Maybe it has now, I don't know, but there was the problem that UniProt kept growing and growing and growing, and we couldn't include those sequences. That's why we moved to Jackhammer, because it gives reasonably well multiple sequence alignments. There's also a method where we realign. We're currently testing if this will give us better performance if we basically use one multiple sequence alignment method to pull out the homologous sequences from the database, and then use another algorithm for making higher quality multiple sequence alignments from these sequences.
I just wonder, does it work for homomeric interactions also? Because so it's the same sequences in both subunits then.
Yes, so for homomeric sequences, what we basically use and have, I probably can't show that anymore. Okay, so what we use is the standard method of, that was published under the name EV-fold. So we just use one sequence, and then we try to disentangle those contacts that are within the same monomer and that are between different monomers. What you need for that is at least some model or a structure of the monomeric subunit. Like if you have one structure of one subunit, and then you can look for all basically what appears to be false positive ECs and try to map those on. But we tried it and it worked. So.
Real quick.
I'll try to. So have you considered reversing the process by doing unbiased docking and then taking the docked solutions and looking for evidence of those contacts that you predict in your alignment instead of the other way around that you're doing it now?
No, we haven't done that.
Because it might be able to get away with fewer sequences, possibly.
Like that could be put, yeah, maybe we should discuss later. That'd be a good idea. Yeah, thank you.
Thank you very much, Charlotta.